So it's my pleasure to introduce Maya as our next speaker. Maya is an assistant professor at Stanford University in the Quantitative Science Unit, which is in the School of Medicine. And she's also in the Department of Pediatrics. She's a statistician whose methodological research is focused on meta-analysis and other forms of evidence synthesis, as well as on causal inference. She's also been deeply involved in work on reproducibility and replication for many years, been involved in a number of the major projects, particularly in psychology. And she works alongside me as associate director of the Center for Open and Reproducible Science, which center we run at Stanford that's focused on these issues. Outside of methodological research, Maya also directs the Stanford Humane and Sustainable Food Lab. And she's received early career and young investigator awards from the Society for Epidemiological Research, Society for Research Synthesis Methods, and the American Statistical Association. So please join me in welcoming Maya.
Great, thanks so much, Russ. And it's really great to be here. It's already been such a stimulating and exciting morning. So what I want to talk about today is some, I think, interesting puzzles that have come up in the empirical studies on replication and what I think this might imply about sort of hidden effect modifiers in science and stability of our scientific results. So I think in this audience, we're all too familiar with scientific evidence that often our findings are not as replicable as we hope them to be. So following a series of high-profile retractions and early empirical efforts to try to replicate published findings, a very, I think, productive discussion ensued, largely about sort of explanations related to selective reporting. And we've heard a lot about this earlier from Edward, right? So obsession with statistical significance, p-hacking by investigators that causes bias in individual studies, and publication bias by what investigators choose to submit or what journals choose to accept, which could cause a kind of missingness not at random in the literature at large. And I think undoubtedly these are probably the most significant contributions to the replication crisis that we have. And so I think this has been a great discussion. At the same time, I think there are still some kind of lasting weird things that have shown up in some of these replication studies that are worth investigating further, and which I think point to an additional role of hidden or what, in other words, unknown effect modifiers. So I think many of you have heard of the 2015 study that went to go replicate 100 experimental psychology studies, where the studies were actually conducted, again, from scratch, using as much as possible the same materials as the original study and input, again, where possible, by original authors. And these replications were powered to detect the point estimate that was reported in the original study. So here's the kind of classic figure that many of you have already stared at, but this is showing on the x-axis the estimate in the original study, and on the y-axis the estimate in the replication. And so if everything were going great, right, and the original studies and the replication studies were essentially exchangeable, then we'd expect these points to kind of scatter randomly around a 45-degree line. And in fact, of course, what we see is that the estimates in the replication studies are typically quite a lot smaller, so actually half as large as those in the original studies. And then the kind of headline result that was most often reported was that 36% of the replications were significant compared to a whopping 97% of the original studies. So a lot was made of that kind of 36% figure. I think arguably it's actually one of the less interpretable metrics that was reported in that paper, in particular because it's actually pretty hard to intuit what the expectation of significance agreement between two studies is. It depends on the power. So it's not 95%, it's not alpha squared or something like that. It depends on the two studies' properties. Possibly a more useful metric of replication success than this agreement in significance is to use the original studies to build a prediction interval for where the replication should fall. So this is basically saying, given the point estimate of the original study, its statistical error, and the statistical error of the replication study, what is an interval such that under repeated sampling should contain the replication study's point estimate? So we can build a 95% prediction interval from each original study. And it turns out that, well, not 95% of the replications are in the intervals, but rather 77%. So that seems a little low. A more recent study did basically the same thing in preclinical cancer biology, which involved replicating 158 effect sizes from 23 high-impact papers in the field. And here, things looked, if anything, even worse than in psychology. So the replication estimates were 85% smaller than those of the original studies, and only 58% of the replication estimates were in the original study's 95% prediction interval. So, okay, what actually, though, should we have expected from statistical theory? So I've already kind of thrown a little bit of shade at the 36% significance agreement figure. Let's focus on prediction intervals. So under the sort of null hypothesis for a good scientific ecosystem, in other words, no selective reporting in the literature, no errors or fabrication of any kind, we might think, well, we built these prediction intervals so that under repeated sampling, 95% of the replication should be in there. So story closed, right? Yes, if we make a key assumption, which is generally left unstated, and this is that the population effect size is actually the same in the original study and in the replication. So what I want to focus on here is basically like, is this realistic? And this is not just a point of kind of statistical pedantry, although I love statistical pedantry, as do many folks here. But I think it actually is more than that. It's really about how we think about, interpret, and ultimately even design replication studies, and how we think about the stability of the scientific evidence base more broadly. Now, unfortunately, this assumption, which we can think of as a no heterogeneity assumption, right? No heterogeneity between the original study and the effect size in their true population effects. This actually can't be verified empirically, right? Because just because we see a discrepancy between the original study and the replications, we don't know how much of that is just the usual casualties of publication bias and p-hacking versus real, genuine effect heterogeneity. So one important potential source of differences in the actual effect sizes between original studies and replications is protocol infidelities, right? So differences in the way that the replication study was conducted compared to the original study. And in particular, differences such that key effect modifiers actually differ in distribution between the two studies. And indeed, some of these replication projects have been criticized, I think sometimes reasonably, on these grounds. So for example, in the psychology replications, there was one original study that was on attitudes towards African Americans in the US, and this was replicated in Italy, where biases and history could be quite different. There was another study that we'll return to later where participants read a scenario that had to do with being in a college class and being unprepared to answer a question that's asked of you. The original study was done using undergrads. The replication was done using online participants. So, it could be that the scenario is just less salient or less important for those individuals. And when we only do one replication per individual study, we really have no idea how much these protocol differences could matter. So I think for this, it's useful to look at the sort of now, I think, quite good evidence base, quite large evidence base from multi-site replications, which is where not only do we do one replication for an original study, but we actually do multiple, and sometimes across the world or across some broader region. And so these designs first were employed at scale in 2014, starting with Rick Klein's ManyLabs 1. Not only do these really improve on the power of single-site replications, but they do also let us do some limited empirical assessment of heterogeneity. So in this first ManyLabs study, this involved 36 multinational sites, which each replicated 13 landmark psychology studies, which together contributed 16 effect sizes. So this is kind of their key results. So the rows are different effect sizes that were replicated, and the X's are the effect size in the original, and then the circles are point estimates from the replication studies. So just eyeballing it, I mean, it sort of seems like, okay, there's like some variation across even the replication studies. Now, obviously some of that, maybe all of it, is statistical error, but we can actually do some analyses to try to look at that. Now, importantly, in I think probably all or almost all of the multi-site replications so far, the replicators have really tried to minimize deviations from the original study. So tried as much as possible to keep the protocol and the materials as similar as possible, though, of course, discrepancies do happen, and also trying to keep the analysis methods as similar as possible. Okay, so remember, the key implicit assumption for many assessments of replication success is that the population effect is the same in the original study and in the replications. And if that's the case, in other words, if any discrepancies between the original studies and the replications are completely attributable to crisis-type elements like selective reporting and errors, then at minimum, we shouldn't have any heterogeneity among the replications themselves, not including the original study, but just the replications. Now, it's not sufficient for this to be true, right? Because it could be that all the replications are getting the same, they have the same population effect size, but it's different from that in the original study. So it's not sufficient, but at least this would have to hold in order for this assumption to be true about the original and the replication studies having the same population effect. So let's check empirically. Okay, so Many Labs 1, so just to give you a little bit better sense of where these replications came from, it was, again, 36 replication sites, 25 in the US, 11 abroad, and then 9 collected online. And as with the other multi-site replications, they use highly standardized data collection procedures and exactly the same statistical analyses. So one thing we can look at, which they reported, is a measure that's sometimes used in meta-analysis of heterogeneity. And this is called I-squared, and it's the proportion of the total variance across the replication point estimates. So that's the variation we can see transparently in the forest plot that is due to actual variation in the population effects. In other words, genuine effect heterogeneity, and not just statistical error. So of these 16 sets of replications, the average I-squared was about 0.4. So about 40% of the variation that you can eyeball from the forest plot is estimated to be due to actual effect heterogeneity. So is that a lot? Well, some people have argued, I think pretty persuasively, that it kind of is a lot, like given the context. This is a context, right, where we're doing the same study over and over using almost exactly the same protocol, and yet we're still seeing what seems like a decent amount of heterogeneity. And for instance, we can compare this amount to what's seen in kind of meta-analyses in the wild in the same field. So meta-analyses are different from replication studies, right? Because they're just a collection of studies that are on approximately the same topic, but typically differ in a lot of ways. They differ in design, they differ in recruitment, they often even differ in what the actual treatment is and even the outcome. So in these kind of in the wild meta-analyses, we do see a bit more heterogeneity, so typically about 0.7 to 0.74. But it's still interesting that it seems like a good chunk of that heterogeneity that just happens in the wild is even present in the multi-site replications. Now, one important caveat here is that I-squared is actually basically an intra-class correlation. And as such, it measures heterogeneity relative to the total variance, inclusive of statistical error. And so this measure actually depends on study sample sizes itself. We'll see something a little bit more concrete soon. Okay, so this I think was very interesting. This was an attempt to directly estimate heterogeneity by synthesizing multiple multi-site replications to date. So multiple projects, each of which had multiple multi-site replications, and looking at heterogeneity for each one of those sets. So I'll focus on the 19 that were on the standardized mean difference scale for comparability. And so here, the average I-squared was a bit lower. It was more like 0.25. What I think is maybe a more intuitively useful metric of heterogeneity is sometimes called tau, and it's the standard deviation of the true effects themselves. So this is, again, on the standardized mean difference scale. That was estimated to be 0.14. So seems like less than in just the Klein Many Labs 1 project. And maybe it's not that much heterogeneity on average, but like here's a thought experiment. So let's say we had a really unsuccessful set of replications. The mean effect size is exactly 0, and the population effects are normal. Well, if we have even this average amount of heterogeneity, so standard deviation of 0.14, then it still means that about a quarter of those population effects are larger than a standardized mean difference of 0.1. That's not a large effect size, but I mean, considering that the average reported effect size, even in the published psychology literature with all its publication bias, is about 0.5, this seems kind of not trivial, right? This is again a case where it's a totally null set of replications with an average amount of heterogeneity. And taking things farther, some of these replications did actually have quite a bit more heterogeneity. So 3 of the 19 actually had a standard deviation greater than 0.3. And going back to our thought experiment, that means that about a third of the population effects are greater than a standardized mean difference of 0.1, and a quarter are greater than a standardized mean difference of 0.2, which is actually sometimes taken as kind of the upper threshold on a small effect size. So, taking this together, it kind of seems like this is potentially not trivial heterogeneity, especially in some of these cases. So even though these multi-site replications do often show relatively little heterogeneity, as we'd expect, it's also not uncommon to see non-trivial amounts of heterogeneity. So where does it come from? Well, some of these multi-site replications have actually specifically gone out and postulated effect modifiers that seem plausible, or even that critics of the replication projects had pointed out, and actually manipulated them to see if they matter. So in Many-Labs 1, they looked at, for example, the role of the mode of data collection. So does it matter if you collect data in person versus doing it online on a crowdsourcing platform like Mechanical Turk, for instance? Also, does it matter what country you do the study in? And they found across their multi-site replications, didn't really matter very much. Okay, another study was led by Charlie Ebersole. And this was looking at a sort of interesting folklore in the social sciences, which is that maybe if you collect data using college undergrads, it matters when during the semester you collect data. Like maybe you don't want to collect data during finals week because people are just really distracted and sleep deprived, so they're going to just respond differently. And so they actually investigated this. They went and did replications at different times during the semester, and they found that it mattered not very much. Okay, another study by Rick Klein, again, looked at lab versus online data collection and country. And then they also looked at task order. So meaning if participants are doing a series of studies one after another, does it matter what order you do them in? And they found that these things mattered not very much. Okay, last one is Many-Labs 5. This one was a little bit different. So here, instead of the replicators themselves trying to think of what might be plausible effect modifiers, they actually worked with the authors of originally published studies that had failed replications in the 2015 attempts. And they worked with the original authors to try to debug the replication and say, well, what do you think we did wrong? Like, can you help us design a better protocol that's more faithful to the original? And as I said, I'm actually sympathetic to some of the concerns that sometimes the studies did have issues that may have potentially contributed. So the original studies were contacted. They worked with the replicators to try to design and approve a new protocol. And this Many-Labs 5 study compared that to traditionally designed replication protocols where the original author didn't necessarily have to approve it. So they looked at whether original study author involvement helped, and it didn't really help. Okay, well, so it's interesting, right? Because it sort of seems like our best efforts to date to try to guess the effect modifiers and manipulate them haven't really improved replication success that much. And yet, we do see empirically some heterogeneity in these multi-site replications that seems like it can only be attributed to effect modification rather than to publication bias. And again, that's not even looking at the original studies, it's just looking at the replications. So I think one striking example of this is a replication sitcom of sorts that I was involved in. So I want to tell this story, which is actually about one of the multi-site replications in the Many-Labs 5 project. So this was a replication of a very interesting study on whether people are reluctant to tempt fate. So here's how the original study went. So it was done on college undergrads. And so the participants were randomized to one of two conditions. So in one condition, they read a scenario saying, okay, imagine you are sitting in a college lecture hall And you came to class having not done the assigned reading. And the professor asked the class a question, and no one volunteers an answer. It's just crickets chirping. And what is the likelihood that the professor chooses to cold call you on your unlucky day when you came to class not having done the reading? Second condition was the same story, but you imagine coming to class having done the reading. So you're prepared. And again, what's the likelihood that the professor chooses to call on you? Okay, so the idea here is that it was hypothesized that people have a sort of superstitious aversion to tempting fate. So coming to class not having done the reading is tempting fate. Like there just might be some cosmic irony where that's the day where you get called on. So I was looking at whether people engage in this type of magical thinking. And indeed, that's what the original study found. Okay, so back in 2015, we attempted to replicate this study, and the replication was totally null. But we did it online. And I think a reasonable concern is that the online participants were not generally college students. And so maybe it's just not very salient to them to read the scenario, or they just don't actually care that much about answering questions correctly in class. And so maybe the scenario is just less salient. And then the other concern that the original authors raised is they did this study at Cornell. So this is a selective place. So it may be that undergrads there care especially much about answering questions correctly. Maybe there's just a lot of academic pressure. Okay, so here's what we did for Many Labs 5. So we did 3 sets of replications. So the primary one was we worked closely with the original authors, and they were really great to work with, like very forthcoming and thoughtful. And so we designed a replication protocol where we would only recruit from universities, so it would all be students. And they would be universities that were similar to Cornell and that they were all in the US and they had similar SAT score profile to Cornell as well. So that's what the original study authors approved. Then we also redid the 2015 online replication, this time with a larger sample size. And then third, to try to get at this question of does it matter how similar the university is to Cornell, we also did it in person at universities that either weren't in the US or were not similar to Cornell in their SAT score profile. Okay, so we also asked a bunch of questions that were essentially manipulation checks. This was actually the most fun part. So fun fact, can you guess? Who said they cared more about answering questions correctly in class? Was it Mechanical Turk users or was it students at elite universities? Well, it was Mechanical Turk users cared more. So part of the hypothesized mechanism of the replication failure didn't really seem true. So they actually cared more by a lot. So unfortunately, we got pretty null results again. So this is the original study's point estimate. And then in Mechanical Turk, we got something pretty close to the null. Our primary estimate was the first orange diamonds. This is the similar sites, and that was quite close to the null. And actually, if anything, the dissimilar sites were a little bit farther from the null, but taken together, it didn't really work very well. Okay, now here's where the sitcom part starts. So we did not know that at the very same time, one of Rick Klein's replication projects was also replicating the same study at the same time. And I also just want to say, I'm so grateful to the original author, Jane Risen. She put up with like 3 different replications of her project, and she was so nice throughout all of it. So they were doing this at the same time. Okay, and as I said, Jane Risen was really very helpful in sharing all the original materials with both replication teams. So the designs were really pretty similar. So we should get the same results, obviously. Okay, let's compare. So the original study, remember the outcome is the perceived likelihood of getting called on. So it was an 11-point scale of perceived likelihood. Original study estimated a mean difference of 1 point on this scale, p-value 0.03. So for us, in our primary analysis, we got a mean difference of only 1/10 as the size, so 1/10 of a point on the scale, p-value 0.64. Okay, big failure. Okay, but Rick Klein's approach worked. So they got a mean difference of 0.54, 5 times larger than ours. Still smaller than the original study, but it seemed like a replication success. P-value less than 0.001. So what happened? Maybe their group is just better at leading replication studies than me, which is possible. So we dug in and tried to compare everything we could about our protocols and analyses. What could possibly explain this? Well, could it be some difference in the way the questionnaires are set up? We found only the most minor of differences. So for them, the outcome scale ranged from 0 to 10. For us, and in the original study, for them, it ranged from 1 to 10. For us in the original study, it ranged from 0 to 10. They had an online questionnaire that allowed people to skip questions, and ours didn't. We did the study in person, but it was people in person in cubicles using a computer, and the original was on paper. For them, the study was part of a larger questionnaire with multiple studies sequentially, and ours wasn't. It doesn't really seem like these things should matter very much, but hey, we actually had a cool opportunity to check this empirically because it turns out we both used Mechanical Turk as one of our data collection sites. And so you'd think if any of these questionnaire differences mattered, that they would play out in this sample. So let's compare. They got a Mechanical Turk estimate of 0.21, and we got a Mechanical Turk estimate of 0.21. So it doesn't really seem like the questionnaire is a great explanation. So let's scrap that. Okay, what about analytic differences? Well, they were really pretty similar. The original study just did a t-test, but we had to deal with the multiple sites. So they fit a model that allowed for heteroscedastic variances. Ours mimicked the original and assuming homoscedasticity. They fit a model that when aggregating across sites assumed no clustering. They just did a t-test, gluing all the sites together. And ours had random effects by site. Now, none of these things should really make the point estimates inconsistent, but whatever, let's go ahead and dig into this. So Rick Klein very kindly let me actually reanalyze his data set before publication and synthesize the two data sets at the individual participant level. So I fit a few different models that made different assumptions where the goal was, glue the two data sets together. So we're doing exactly the same analysis and actually empirically estimate the size of the discrepancy between the two sets of findings. Well, doesn't really matter what model you fit, you still get about a 0.4 point difference between the estimates. So we can kind of scrap that. Well, definitely the most important protocol difference was the sampling frame. So they recruited undergrads at various colleges throughout the US and abroad, versus again, for us in the primary analyses, it was only US colleges that were similar to Cornell. But conveniently, right, we did also have these dissimilar universities as well. So what happens if we add our dissimilar sites to the analysis to try to make the sampling frames more comparable? Well, this slightly attenuated the discrepancies, but didn't eliminate them. So that's the point where I didn't know what to do anymore. Despite my best efforts, we really don't know why this happened. And I think given this, it does seem kind of hard to escape the conclusion that there were some actual hidden effect modifiers here that we just don't know what they are. We talked to Jane Risen, the original author, and she was like, I don't really know either. Unless there were actual errors in data preparation that somehow affected only the non-mechanical Turk sites. So I don't have an answer here. I do think it's interesting to look at a different study, which takes a very surgical approach to diagnosing a replication failure. This was led by my colleagues Molly Lewis and Mike Frank, and this is a replication of a study about the role of social context on generalization during word learning. So the idea of the original study was basically that, okay, if you're learning new words by seeing examples, you have to sort of do an induction from the examples to some general concept, right? Like if you're trying to learn like what a border collie is and you're just seeing a bunch of dogs, like you have to decide, does border collie mean border collie? Does it mean all dogs? Does it mean like just a certain color of border collie, et cetera? So the idea of the original study was that the extent to which you generalize from your examples depends on whether you are just encountering the examples in the wild versus if someone is explicitly teaching you. And the gist, I think, is basically that if someone's teaching you, they're going to choose examples for you that kind of span the whole class of possibilities. So the original study that was replicated by Zhu and Tenenbaum manipulated whether participants were learning words from a teacher or not from a teacher, but holding constant the actual examples that were given. So it's just about the role of the teacher, the social context of learning. So they did the study in both children and adults, and either way, they got a pretty huge effect size by social science standards of 1.2 to 1.4. Now, this is a pretty imprecise study, though. These are very wide confidence intervals, but point estimates at least were pretty large. Okay, so Lewis and Frank decided to replicate this in an online paradigm. Seems reasonable. Compared to the original study was done in person. And the replication study had a way, way smaller effect size than the original. So 0.17, confidence interval crossing the null. Okay, well, here's what's cool. Then they said, let's try to think about what could have gone wrong here. Well, when we do the study online, the teacher role just turns into like a block of text where it's like, I am teaching you blah, 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 but it's just like text that you read, as opposed to a physical teacher standing there with you in the room. So they said, maybe we need to make the role of the teacher more salient by actually having a picture of this person. So they did that. They did a new replication study, and that somewhat increased their point estimate to 0.33. Okay, but then they still weren't done. They said, all right, let's actually just do in-person data collection with a live teacher, just like the original. So now they got an estimate of 0.45, again, a little bit of an increase. So that was the most they could do with making the teacher role the same as the original study. Then they said, well, maybe we need to make the actual stimuli whose names people are learning as similar as possible. So it was like these little shapes with colors inside. So let's use the same shapes and the same colors and exactly the same stimuli as the original. And that actually also increased the effect size a little bit more. Okay, then having done all of this kind of principal cherry picking, they finally did a pre-registered replication of this final experiment with a larger sample size and ultimately got an effect size of 0.71. So also, I just love, look at this beautiful monotonic increase in point estimates across the replications. Like, isn't that so cool? So over the course of these 5 replications, the estimate increased from 0.17 to 0.71 with a confidence interval bounded well above the null. So that's pretty interesting. And I think it really, again, points to the role of effect modifiers, which in this case, actually, the replicators seem to have guessed some of them correctly. And I think one thing that's interesting here is that the replicators in this case are actually domain experts in the field of language learning. And so it seems possible that they were sort of maybe more able to figure out what the effect modifiers are than in some of the previous replication studies. That said, the final estimate is still quite a bit smaller than the original study's estimate. So how much of this is just the usual issues of imprecise original, selective reporting, et cetera, versus maybe there are still effect modifiers left that even these domain experts weren't able to guess and manipulate. So just to wrap up, again, we saw that multi-site replications that use highly standardized protocols and analyses do typically have the low heterogeneity that we'd expect. But we do also see pretty regularly moderate to high heterogeneity given the context as well. And it doesn't really seem plausibly due to selective reporting if the replications are pre-registered and pre-accepted or to analytic differences. And yet, a lot of our best systematic efforts to date to try to manipulate speculated effect modifiers haven't really come up with all that much. And really one of the few exceptions I'm aware of is this single case study of Lewis and Frank, which is the only one, again, involving repeated replications. And so I think, hidden effect modifiers may be more common than we realize, and it has implications for how we design and interpret replication studies. So I think first we should be pretty wary of assessments of replication success that implicitly assume no heterogeneity, like the kind of significance agreement figure, or even standard prediction intervals. It is possible to extend these approaches to impute a certain amount of heterogeneity. And that makes sense because if we don't, we can actually end up with too conservative estimations of replication success. I think it's also great that there's been increasing prioritization of doing multi-site replications because with only one site, it precludes really all estimation of heterogeneity. And last, I think it's reasonable to start focusing a little bit less on replicating individual studies and their specific protocols, and rather to kind of embrace the heterogeneity in the design of the replication studies at the outset. So perhaps replicating distributions of protocols and effect modifiers that are seen in meta-analyses, for instance, rather than saying, well, we're going to look at this one very specific operationalization of tempting fate, for instance. And all of this has been about multi-site replications, but I do think it has broader implications for the way we think about generalizability and transportability in science at large. Typically, when we're not doing a multi-site replication where it's a perfect petri dish of highly standardized protocols, we might actually expect effect modification to be quite a lot more across plausible contexts. And I think this may have sobering implications for how well we can generalize and transport effects across studies. And in particular, when we have unmeasured effect modifiers, we usually can't get a consistent point identification in these types of contexts. So I'll leave it there. I think I actually have more questions than answers myself, but I'm happy to take your questions, or maybe you have answers for me as well.
Thank you for a super interesting talk. So going back to your first case study, right? Did you guys have data in the replication about who are the students who in these different universities, right? But it's not just the university, but you might, how big are these studies? Like 100 students? So do you have characteristics, what majors, all of that to see? Seems like mechanical Turks for that aspect, they seem to be more similar than, I mean, you have a finite population of 2 studies. How big were the studies? Like,
yeah, it's a great question. So the similar sites combined had about 754 participants, and each individual site was anywhere from 50 to I think max was like 200 or so. So it was a little bit hard to look at individual site variation. We did ask some questions about things like demographics. We asked these questions about, how much do you care about answering questions correctly in class, and nothing really seemed to pan out as a clear effect modifier. Now, demographics do differ a lot on mechanical Turks. People are a little bit older, typically skew a little bit more male, and more geographically distributed as well than the sites we use.
so couldn't find anything there to explain.
Not really.
Thank you.
Quick. So fantastic, really interesting. So I have 2 questions. One is small and one is big. To what extent, a lot of, I didn't track what the effect measures were. I saw it was Cohen's D. But a lot of the quote unquote effect measures in psychology seems to be statistical measures that depend on underlying variability, which I never expect to stay constant, as opposed to like a mortality rate or number of minutes, you can run a race faster. So how much of this was due to statistical outcomes that really we fantasize should stay constant, but can be just an artifact of other aspects of the sample? And the second one, which is closely related, and this is a diss on maybe on the whole field of psychology, which is, if these theories are so fragile that tiny little changes in design can produce these big, changes in effect, whatever that effect is, are they useful for anything? I mean, how can you generalize them to any context to actually improve, whether you're studying teaching or anything? Because you have to have the shapes of the of the figures you show to be a certain shape, or the podium that the teacher stands on to be a certain height. So, if you see such fragility, does that indicate that the basic theoretical framework of the discipline or these hypotheses is too weak to be useful?
Yeah, these are great. So second one first, I totally agree, honestly, and I think this is partly why. So honestly, I actually still kind of believe in the tempting fate effect. Like, so does Jay Rice. I think it's probably smaller than originally estimated, but I do think the hyper-focus on, replicating perfectly copying this one protocol isn't really serving us that well, and that's why I would prefer to see replications look at a distribution of reasonable operationalization, where sometimes that reasonableness is going to interact with the population, like in this case where I think she did credibly argue, like, it doesn't make that much sense to use this scenario. Like, her research is built around this effect. It's not built around the sitting in college class question, and yet sitting in college class question is what we replicated. So I totally agree. I would love to see approaches where, for instance, you take a meta-analysis, you look at the types of protocols done there, and you try to replicate those. There's been a little bit of work starting like that, but I do think that's going to be critical. And then to your first point about kind of effect measures that differ inherently across populations, I also agree on that. I think it's especially a concern when it's something like a standardized mean difference, which we see a lot of in psychology. They can be useful in context, but if you're dividing by that standard deviation, so you get a less variable sample, and then you get a larger effect size. That's why, like, when I was reporting on the one I was able to analyze, it was all absolute mean differences. Now, you still could argue, well, maybe the size of a meaningful effect on this likelihood scale depends on the context, but at least it is an absolute measure. So I agree, and I think often when we synthesize the multi-site replications, everything ends up getting converted to a correlation or a standardized mean difference, and it's not ideal. It's a problem that's there in meta-analysis too.
Okay, we have time for one more question.
Hi, thank you for your talk. It was very interesting. I had 2 questions kind of correlated. So for the first one, you mentioned how a lot of replications have to make adjustments. Kind of, you gave an example of using an online survey versus in person for the Cornell example, and then like different metrics were used. So I guess I wanted to ask, at what point does a replication stop being a replication? Like when these differences just accumulate to the point where now you're answering a different question, whether by choice or not, just the data availability and such. And then second of all, kind of bouncing off of that, is there an objective metric to gauge whether a replication is good enough, like how close it is to the original, aside from comparing the results of the replication with the originals? Because like you said, we don't know if if their difference is due to the originals being like imprecise or the replication not being accurate enough to the original study.
Yeah, this is a hard one. I've been struggling with this too. So, I mean, I think to the first point, I guess I've been starting to think of it in terms of like differences between the original and the replication that are varying on what we would theoretically think to be nuisance parameters. So something like time of semester or maybe even the college where it's conducted, like kind of feels like if we're trying to build a sort of relatively universal psychology of human cognition, for instance, like shouldn't matter all that much. But then you could think of things where it really seems like this is actually of importance. Like so actually in this study, Tempting Fate, the original study also had a manipulation where it gave people a task before the study that made them frustrated or not. And so it's like this interaction of how frustrated you are. Maybe if you're more frustrated, then you engage in more magical thinking. So that's like a substantive effect modifier, where it's like, okay, maybe if we do it with participants who are frustrated for some other reason, that actually contributes to the scientific theory more so than undermining it. So I haven't really seen that hashed out in the actual design of replication studies, partly because it's just limited power to really manipulate all these things. But I would like to see us think a little bit more in terms of, what are the effect modifiers that are of scientific interest versus nuisance ones, and ideally kind of pre-specifying that as part of the theory. Like, what are the contexts where it should generalize?
All right, let's thank Maya again.